Hi, this is Eli Ocheltree of the SnoKing Beekeepers Association. It's our third Wednesday monthly meeting, week before Thanksgiving. So we're going to go ahead and have the meeting, and we will record the most important parts. And the discussion afterwards, we don't need to. But for people who can't make it with all the snow, well, snow is supposed to follow now, possibly. But right now it's the power outages after the wind damage. So that's why we're recording, and we tried to record anyway. I I have a personal plea here. I really wish it would freeze. My bees don't understand they're supposed to be going into winter mode. Everybody, public service announcement here, check that they're not eating up all the stores that you had put in that hive, because this is on the upper right, it's the graph I love to show that explains why sometimes your bees run out of food and sometimes they don't. And this is the kind of winter where it's too warm. It has still not frozen where I am. That line, it says colony metabolic rate. Basically, that means how much food they're using, a rough translation. And if we could stay down below 50 ambient, below 50 ambient, if we could get to 45, it keeps going to the 50s. And then the bees at night times, even, it's still in the 40s frequently around here. Depends on where you are. So watch your bees, because if they would just go a little colder, they would stop eating their stores. And I am pretty sure, looking at the weights of some of my hives, that they have eaten all their stores up. And that is part of why we are going to talk about soft fondant. Because you're saying, why bother to make soft fondant? Just dump some sugar in there. Well, there actually are a couple of reasons. But this is what scares me, is I look at these temperatures, and I wish brood rearing would stop at 50 degrees. Over on the left-hand side, do you see where it says that 50 degrees brood rearing stops and workers can't fly? Ha, ha. We know they fly. But the problem is they're using up a lot of energy, can't fly far. I think the 42, they can't move. Their muscles just aren't warm enough. 45, I have read in a lot of places, and it makes sense to me. They can fly if they keep moving. Don't stop and get back to the hive, not without stopping. So I just put that up because I really, really want it to freeze. And this year, it is not freezing. It is still above average temperatures, despite the winds and everything. We do have another in-person social. If you looked at the newsletter, you saw those happy, well-fed faces. The next one's going to be January 11th, and Jen Murray's going to host it up in the Kenmore area. It's going to be kind of rotating around to whoever volunteers their home. If you'd like one in your area, or maybe you could talk a beekeeper in your area into it. We're trying 3 short topics tonight instead of one big presentation. And we need more of those sorts of things where people don't feel intimidated as if they have to come up with a whole hour of talking. A lot of you do-it-yourselfers. I think we should have just one on do-it-yourself or neat ideas that people have. Any kind of presentation, if you are journeyman or close to journeyman, you have a presentation you'd like to try out on a small group, even if you haven't actually taken the journeyman class, I would let you in if you were going to do a presentation on something you wanted to try out on people before going big time and doing it like at a monthly meeting or something, or your home club. We still need lots of articles for the newsletter. You get service credits for that. 2 of your 35 journeyman service credits per article. And I want people to start thinking about the SnoKing's digital library project. There are a lot of older books. I can't remember if it's 50, 75, or 100 years. Once they pass that point, then they are royalty copyright free at that point. The Gutenberg project has some of them. I remember I went through the Internet Archives. I downloaded a lot of them. And this winter, I'm really wanting our club to host a page where you can go and download a PDF. If you go through the Gutenberg project, it takes you hours to go through and try to find everything on bees and apiaries and such. And I think I got almost everything out of there and downloaded it. Internet Archives, I'm not sure if I did. And there's a number of free, copyright free, nothing that we're not doing anything illegal. But if we could put it in one place, wouldn't that be a great idea? Sort of idea that I wish I could get WASBA to do. This Saturday, if you have an urge to go to do a few hours of answering questions about bees, I'll have to put Heidi's contact information in the chat, or you can contact me. It's this Saturday only. It's a Christmas craft fair. And we do need more help on computer, on YouTube, the website. And sometimes it's proofreading that could be done at home, on it, especially if you have any computer access at all. So let me know if this is the time of the year that you could volunteer more. A lot of things don't really directly qualify for journeyman credit, except if you help significantly, I can put you down as a board of trustees member, and you will get some credit for that. Oh, and this just happened to come right before the meeting today, that Bri Price is sending out her emails, and I guess this one's on, it takes you this link to the Apiary Inspectors of America. And apparently they're going through the honeybee pests, and they're starting with Tropicalaps, which is the new mite that's supposed to be worse than Varroa. So getting actually into the meeting tonight, I figure if we do 3 topics, I figure it's gonna take us an hour. I want to talk about the soft fondant first, because that's kind of the topic that's very time critical. If you're thinking about ever doing fondant, you're probably thinking about doing it now. The PubMed database is near and dear to my heart. People struggle with Google Scholar, and I think Google Scholar does have its advantages, but I'd like to show you your taxpayer, you paid for it already, your taxpayer paid free mega database, and the part of it that applies to beekeepers who want full text articles on beekeeping. And Gary, I don't know if Gary came on or not. Oh, he may not have any power over there on the peninsula. But Michael Duncan's here and he's got power. Okay, but he volunteered his report results from the National Honeybee Disease Survey. And if you don't recognize that name, Bri Price was asking people if they had 8 hives. I think it was 48 hives was what they were looking for in apiaries throughout Western Washington. And she would come out and take some samples and take a quick look at your hives and write up the results, send it in to this national survey. And I'd like to go through that and talk about what they looked for, what they didn't look for, what they found, what they didn't find, and do that. I think it would probably take me 20 minutes. If you look at it, it's a piece of paper and you go, well, so what? Unless you're used to reading these, or you're really fascinated by how the research data is really collected. So Colleen made up all the PowerPoint slides for the soft fondant. I'm going to take you through the PubMed database really fast. I'm going to do it in slides. I can also do it live if anyone wants to, but something always goes wrong when I try to leave the slideshow and then come back again. Yeah, I'd better just show you some slides, and then I'd be happy to run anybody through why I love this database for getting full text articles instead of hitting that paywall constantly, and getting the best types of articles too. Doesn't have everything on honeybees, but has an incredible amount. And then that survey. So I'm thinking 10 minutes, 5 minutes, 20 minutes, and then we can just talk. Okay, the first question is, okay, you can just dump the dry sugar on white, right? I mean, why bother with this? You cooking for the bees. I mean, unless you want to do it for your cute little fuzzy girls. Okay, it sounds kind of ridiculous until you've been beekeeping a few years, and then you go, well, yeah, I could take the newspaper, lay it on top of the top frames in the top box, pour the granulated sugar on, make sure I don't block the hole in the inner cover because I still want some insulation, some ventilation through the hive. And maybe I've got my moisture shim up above. Okay, so you don't want to block that hole, but you can take a pretty good sized piece of newspaper, quarter of a page or maybe a little less, lay it down and sprinkle granulated sugar. Somebody told me sugar cubes if they fit. That is part of the reason for soft fondant. Soft fondant is like icing. It smooshes into wherever you want to put it. If you want to set soft fondant, on your tops of your frames, your top box, you can kind of, without putting a shim in, kind of smoosh it down between still on the paper, smoosh it down, and you can still get your inner cover on, or you can put it above. We usually hit a cold time of the year right about now, and the bees can't get above the inner ceiling. They can't leave the cluster. This year, my bees a lot of them are above the inner cluster, and I think they're running out of food. So I'm very interested in this topic. So it smooshes in well, and granulated sugar, if the granulated sugar isn't used in the spring, how many of you have spilled it all over? It's all over, attracting ants and all over, falling through the hive, and is sometimes getting onto the ground. And okay, it's a mess, and you can't reuse it. Soft fondant is you can still pick it up if you put it on and they don't eat it for some reason. You can pick it up and you can, I don't know if you should put it in another hive unless you're absolutely sure that you're not transferring any diseases, but you could add water and make it back into syrup. The the reason that Colleen got really interested in it and wrote the following slides, is she was reading, and I've read it too, that the honeybee mouth, we think of it as a tube and very durable. No, actually, when a honeybee makes its proboscis, it takes about 3 parts, mouthpieces, and puts them together and then forms this little tube that it can siphon up and can regurgitate back out. It's a straw that works both ways. But parts of that straw, look at the little hairs on there. Those hairs, if they have to work with granulated sugar, now they will bring the water over and put the water on the dry sugar. If you didn't make them fondant and provide a lot of the water for them, they're going to have to bring more water over, and then they got to kind of work at getting it up. Supposedly, and I don't have scientific research paper evidence for this, this is really hard on their mouthpieces, actually. And it could be a little bit of a difference in how healthy your bees are in the spring. I don't know if anyone has a research paper, put it in chat or tell me, okay? But that's the reasoning, one of the reasonings behind soft fondant instead of just dumping dry sugar in there. Okay, this is where Colleen got it from. She always gives credit, which is really great. And there's the final product, which looks like a glump of slightly thick icing, rolls like a cookie dough at the end. She has all this, and she has this in a document and also in a PDF of these slides. So I'm going to post those on Facebook. I can also email them, and Colleen would love to email them out to anybody who wanted them. But I'll put them on the Facebook under files. So you get your stuff together. You don't need a big fancy blender. If you've looked at this and Keeman Reynolds did it, I looked at that. It depends on how much you want to make. I've been doing this. This thing works perfectly. It doesn't turn into a hard brick on you, which is what this has happened in the past when I've tried other people's instructions. So she writes it all out, what you need. This is called an immersion blender. It's a handheld. She recommends the cordless just in case, but actually you only need to go for a few minutes. No, the corded is what she recommends, but the cordless does work. It's because you only need it for a few minutes, but maybe you're making several batches and might need recharge time. Okay, so here it is. And this is on the slides, and it's on her handout that I'm going to post under files in the Facebook discussion group. Here it is. She uses additives. She uses vinegar. Vinegar is a little controversial. Uh, some people put it in. There's two different reasons people put it in. One is to start changing the sugar. Acid changes sugar. It starts some of the breakdown into, I guess, the fructose and glucose parts of sucrose. And then the other reasoning is that if it's slightly acidic, it's better for bee stomachs because it's closer to the acidity of honey. Take your pick on if you believe in it. I don't think the vinegar is at all necessary or any of the additives, but this is a good time to put them in if you're going to try them out. So here's the how many pounds, how many cups. You're doing 4 to 1. And then just up the vinegar, 1/4 teaspoon for every 8 cups. Measure it. You do have to have a pan that will let it boil. You're not going to stir it down. You're only going to stir to mix it in, not dissolve, but just get all the sugar wet. That's all you're going to do. Then you should have a candy thermometer, and Colleen says the glass ones don't work as well as these digital, so I trusted her and bought a digital. They're not expensive at all on Amazon. And she says, hang it with this, should have this clip, and it's adjustable, it slides up and down so that the thermometer does not hit the bottom of the pan, it's a little bit above. Put it on the burner, you turn it on medium high. Now this is the part that is not the same as the other ones that I tried. You do not stir. If it's boiling up, I've knocked the bubbles down a little with the spoon, but it shouldn't boil over. You're bringing it up on medium high, up in temperature. Do this 23.4, and I think she says 0.5, 234.5. Okay. It'll start getting clear, but you still keep going until the syrup is up to 234.5. Take it from the heat. You can just lay it on, put it over to the side to cool, not stirring still, or you can put it in a cold water bath. This is, you've gone above the boiling temperature of water. Be careful not to splash any water if you're going to put it in the sink, for example, because the water's going to boil and splash up immediately into your face with the syrup. So be careful if you are going to use the sink. Okay. Now as you're cooling it down, still with the probe in there, your digital thermometer in there, then you wait till it hits 130 degrees. And that's when you put in the vinegar, any additives you wanted, and then you kind of go up and down. She was introducing air that way. Just kind of trapping some air and going up and down. And it turns white very quickly. It really is. It's really, really fast. About 2 minutes. And it sets up really fast. And she had to do it by hand. She says she can do it by hand. Good muscles there. Then the only problem is you have to stay in there because you have to be right there to do some of this stuff. You immediately transfer it, scooping it out with a good strong spoon onto parchment wax paper. And paper plates are wonderful because it lets it kind of contains it, a mounded pile, and then you can push it, mold it a little bit. You can use another piece of paper. When you go to store these, you're going to need paper on the top and the bottom to keep them from sticking to each other and even to the plate. So that's why she says store it unused in Ziploc bags and freezer. I was going to ask her if you could, if you could put it in the freezer. Yes. So you can make this on a day that, hey, it's so bad weather, you can't go out and do anything else if you want. This is super easy. It works. It stays soft. It's moldable. Smooshes into whatever space you have, so you don't have to buy fancy shims or anything. If you need to put it under the inner cover for the coldest part of the winter, or put it on top. That is actually what she had to say. Oops. Is there a quick question, or should I just go on? And I'm going to be sending this out. I'll post it by PDF, And then anyone wants to can email me or I can put Colleen's up too. And I know other people have fondant recipes they might want to discuss, like at the end. Is it okay if I just go on, assume that this has gotten you thinking, and you might want to talk at the discussion at the end about your favorite recipe, why you would do this, why you would never do this. But I gave you the 3 reasons that I know of why people do soft fondant back at the beginning. I'm going to take silence as consent. And oh, I really want to do, I want to leap ahead to this one is going to be fun. This is Gary Heller's report. Okay, what I have to discuss isn't as much fun as that, but I want 5 minutes to tell you about my favorite database. And since I'm sort of the person with the microphone, I guess I get to tell you. I did research for a while, and this free taxes paid, that's why it's free, air quotes there on the free. You might as well use it because you've paid for it. It's the National Library of Medicine under NCBI, National Center for Biotechnology Information. And this is a part of the database. Okay, the huge database. Genomic sequences are stored in this database. If you look at the left-hand side, it's overwhelming what's in there. Mapping, all kinds of stuff about proteins, domains and structures of proteins. There are libraries on everything. How you do sequence analysis is all kinds of stuff. But the part is honeybee people that we're probably interested in is how do we get a good research paper? How do we get the full text? How do we even know which research papers to get? And then your first question is, what in the world does medicine have to do with biotechnology? I mean, and bees, a lot. Bees are a research animal, and I think I already bored the troph people with that at one meeting, that bees have all the organ systems of people, all the basic organ systems. Nervous, they do have a heart and a circulatory system. They have a digestive tract, an immune system. They have part of what we have, and we actually study bees and fund studies in bees for medical purposes that apply. It will apply. A lot of the information applies to humans. Okay, if you get to this page first, and I usually just type PubMed in and I get directly to PubMed, but I don't know if that's just because my computer's so used to me searching PubMed. If you were to put PubMed in, it probably would come up right away. PubMed. So you select out of all this possible, the left, woo, there's a lot of stuff there. On the other side is sort of the popular resources. PubMed. So if you select that, click on that, you come up with this, and this is what I'm used to looking at whenever I want something. And again, I said if you put PubMed in first, it'll probably complete that address that's up there. And you get to a search bar. And then it says login. Ignore login. You don't have to do it. You want to set up an account. You get a free account, and I think it does keep track of things for you. I haven't done it in years. Okay, you get here. You want to look at bees. You could put in honeybees. I would put in Apis mellifera just because I figure that'll get me there faster, more specific. Well, it's not that specific. How many papers do you think relate to honeybees? Books, articles, papers, everything, do you think are in this database under this fraction, PubMed, of the entire database? And yes, if you want the genome of the honeybee, it's in the other parts. Okay, 24,081 results. Okay, that's useless. That's kind of like you go to Google Scholar and you got too much and you don't know what to do with it. But this is where it outperforms Google Scholar. Because Google Scholar has a filter, but it's so hard to use. I've never really gotten the knack of it. Again, ignore the login. All you did was put in honeybees, Apis mellifera, and you got 24,000 results, but it's way too many. What we do is over on that left-hand side, did you see where it had this? And there is a whole set of filters down there in a nice, easy to read list. You can see it a little better in my right-hand picture. It starts out with results by year, and you can ask for, it tells you how, oh, that's interesting. Between 1857 and 2024, each year, the number of articles about honeybees has gone way up. Gee, isn't that interesting? Way up. That's useful because sometimes you want to select this first category. Do you want just things that were published in the last year? You're looking for something you know somebody just published in the last year or the last 5 years. I didn't do that on this sample, but you could right there say, I know I only want something about the bees and I know it's in the last year, 5 years, 10 years. Or you could pick a custom range. There's actually a custom range too. What I did though was the thing that's most important to me. Whenever I go to like Google Scholar, I get so frustrated because I want the text. It isn't going to be enough to read the abstract. I don't want to pay for the text. I want the free full text. So I clicked on this free full text. That 24,000 dropped to 10,861. Okay, that's still too many. Well, now I'll tell you the secret that most people don't tell you when you do research. If you're going to do any kind of research and you're going to look at research papers, you don't want to just look at generic research papers. You want the paper that's called a review. So I clicked free full text, that stays clicked, and now I click review in that column on the left. Suddenly it drops to 667 results. There are a review. A review article is the article you want when you start looking at a subject. Unless you know you're looking for a specific author, a specific paper, specific title, something like that, you want the review. The review article is the authors are talking about, like if you look up for a review article on oxalic acid treatments, that's useful to you. Thousands of papers on oxalic acid don't help you until you know where you are, what is the body of literature that you could get. So that's what they do. They don't review one paper. They review a whole bunch of papers and they talk about it. And that is extremely useful to you. It's the first thing you should do. Somebody says, go look at probiotics. I would add probiotics actually to APHIS mellifera, probiotics, and then you would get way down. I didn't even add a second term. APHIS mellifera, I got the review articles right away. Maybe you did want to put that in. And I want you to notice that you got from 24,000 down to 667. It's still too many. But we can continue on. I put in queen. Maybe I'm interested in just whatever they're putting out there right now about APHIS mellifera queens. 54 results. I'm going to get free full text, and I'm going to get review papers that talk about other papers, and I can, out of that, select the ones I actually want to read. It's like getting little descriptions like you do on YouTube or some video service, and they tell you a little bit about each movie. Well, this will tell you a little bit, and this one did this, and this one, but this other one did it a different way. Okay, so Queens, I'm down to 54 results. I can read 54 results by reading the titles. I could also add another word. For example, maybe I only care about supersedure or replacement or pheromones or something like that. I could put that in and drop this 54 down. I could say I only want the last 5 years because a lot of things are changing really rapidly in 5 years. This is a great tool to play around with. Sometimes you want to author. You want to know, okay, so I did this because I know that I won't get very many hits. This is me. This is how you search for a person in here. You put the last name, this is standard, and then you put the initial, if you know it, the first initial. You won't get a lot of Ocheltrees, but I put me in because I knew you'd come up with these 5 results. And they do happen to all be free articles because they were done under medical research, not just biotechnology, but medical. So they definitely are going to show up. They have nothing to do with honeybees. It's what I worked on in immunology, and That's why I talk immunology sometimes. And I was in the genetic component of this. Some of these I was doing cut and paste. Yeah. The bottom three are cut and paste DNA to express a gene, overexpress it, and study its effect. And not really interesting, except if you want to verify that I ever did do research, here I am. Okay, this is me. I think that's kind of boring, but I did want to put this into a recorded presentation because, okay, now I'm going to have to go back to Gary's. If there's a quick question on it, I'll answer it. If not, I'll leave this here, and it would really be fun to, I turned my captions on accidentally. I have no idea how I did that. Oh, let me turn the captions off unless somebody wants them on. I don't know how to turn them off. I don't know. I have no idea. Okay. I don't know if everyone else is seeing captions. I'm seeing captions. But I want to go back now because that was just the quick why I like that. I love the filter on it. And the filters are there in Google. I just find them a lot harder to use. Anybody, anything you want to say quickly, or should I buzz on with the recorded part and do topic number 3? You have to unmute and tell me because I can't really see chat if you would. Otherwise, Gary was really sweet. I don't even know if he made it here today. Did Gary make it here today? Because I'm doing his. Okay. I don't see Gary. Colleen couldn't make it for her section because she's out of power and she's been sick. Okay. This was the citizen science study opportunity, one of the ones that came up in this last year. And Bri Price, our honeybee outreach coordinator through WSU Agricultural Extension, and I think her job was partly funded by the Washington State Pollinator Task Force, that if you've been active in WASBA, you've been hearing us talk about for at least 5 years. Tim Hyatt's been working with them that long. He's been our legislative liaison for a long time now and active helping out WASBA with that. This is the results of the apiary visits that they were making, just one person, Gary Heller. Michael got visited too, but I got Gary's up on slides. I don't remember if I ever got Michael's up. I don't think I got yours up. Michael Duncan can also talk about this, having somebody come out, look at 8 hives, inspect them, decide what they saw, and they had a checklist. And on the report, there's things they looked for, things they just quickly looked for, things they were not interested particularly looking for at this time. So there are things that they can compare it to a national average. They can say what the prevalence is, how often it shows up in hives throughout the United States. And I believe there's a note in here about the average results over the last 5 years. So they put you in a percentile, 50% of the hives also had this. You're in that, or you have less than the 50%. You're lower than the national average. You're in that lower part. Okay, I'll try to explain it the best I can. This is what what Gary got on his report. And he's over there, gives his address, in Port Angeles. They were willing to go across. Bri was volunteering to do that. And she also had somebody else who was helping her on some of the inspections. So Animal and Plant Health Inspection Service, you might remember them as the place you send your apiary registration to the State Department, the WSDA, Animal Plant and Healthy Inspection Service. Once a year, we register our hives, and you send that form in. So their purpose is they are monitoring for invasive threats, think AAA lapse or something like that, it's expected to come in, and they document known diseases, parasites, and pests. And by documented, is it there? Is it here? How often does it show up? So, as Gary voluntarily had his apiary sampled by the state apiary. Well, we don't have a state apiarist. Only about half the 50 states, I think it may even be less, have an actual true state apiarist. So Bri Price kind of filled that in. And any results not included in this report, it might be another report later. And this is going to be what visual field inspection, what Bri saw. She sent some samples into a lab for diagnostics. She did some what's called molecular diagnostics, but they didn't check for pesticides. So what she saw visually, what she sent into a lab, and then and molecular diagnostics, running molecular analysis. So this is kind of fun to get back. Okay, these are Gary's results, and it starts out with how many colonies are in the apiary? 11. They sampled 8 of them. All 8 were queen right. If you kind of follow down the lines here, 1 had queen cells. Nobody queenless. These are good zeros. I like 0 is an important data value. Never be afraid to fill in 0. They used to tell us that when we did data collection. No colonies with drone laying queens, no colonies with AFB, no colonies with EFB, no colonies with sac brood, chalk brood, parasitic mite syndrome, deformed wings, black shiny bees. That can indicate viruses. Small hive beetle, they're a little worried about that. They're on surveillance for that. Wax moth, we don't usually have inside of our hives. It's always by stored equipment that it shows up in. So zeros are good. That was good. Okay, now, so all that was good. I put it in larger type in case you needed to look at it again. These slides will be in the recording. If you want to come back and look, zeros are good. I guess I put that in 3 times and it's important to mark 0, even in your hive journaling and such. It wasn't there is important. You didn't forget to put it in. My chemistry instructor used to say all the time, 0 is a valid data value. Put it in. She got tired of people leaving it blank because he felt, well, 0, 0. Okay, so for the diagnostics, what she sent in they did the sampling of the varroa, and Gary did great in terms of varroa load. He was way down here. This is percentile compared to other positive samples. Okay, so compared to other people who they detected varroa, which is probably just about every hive you've ever done, and he's really low. Nosema, it's there. They're actually not that worried about Nosema. And then we don't have any eastern honeybees. Apis cerana? Oh, Apis, no, that's eastern honeybees. And Tropolaelaps they didn't expect to find, but they're looking. So ND, not detected. The key is all important. Not detected, not detected. And counting all the mites in the hive, no, that was not applicable, NA. It's important to read these keys. And they are watching. One thing this report is telling you is that they're starting to watch for Tropolaelaps. They figure it'll come in. We actually kind of expect it to come in through British Columbia from those hives, because those are coming from New Zealand, which is not being careful, and other areas of the world that are not being careful about Tropolaelaps. So it explains. I made it larger in case it's easier to see, because, okay, we're looking at NA, not applicable, and ND, not detected, but it tells you they looked quickly. They either looked or they were looking. So that is actually what you call a 0 value, except they didn't look really hard. How to interpret the data values. Anything else that was important in there? No, I just put it up bigger so if people wanted to be able to see it when they look at the recording, they can see. The notes are everything often. And it's over the one thing it does say is this is over the last 5 years. So if we had a surge or something, this would put note number 2. Reference population is a data set used to compare your pathogen load result to other sampled apiaries in this study. within the same month as your collection date, averaged over the last 5 years. And lower percentiles are good when you're looking at pests and pathogens. Okay, I was just worried. This was a larger, you could see the page better. Now, one thing that was interesting was they looked at Nosema serrani, which is the Nosema that's replacing APHIS, and they consider it more invasive. They consider the national prevalence of it to be at least 66%. And this thing, the pathogen load, sounds terrible, except that by itself, when you say 10 billion copies per bee squared, they got it. I don't know why they're squaring it, but that's the way they are doing it. They didn't try to compare it to other positive samples because I think at this point they're still trying to understand Nosema serrani. It doesn't outright kill your hive, basically. It contributes. It's a factor. It was considered to be a factor in colony collapse disorder, but not a lone cause, not the strongest cause, not even a super strong single cause, but it was there. And they are, let's see, this also happened to be next to slow paralysis virus, not known to be in the United States, but you notice there's an ND there. They are looking for it. They got it under surveillance, even though it's not a virus we're interested in. And it wasn't applicable because it's not in the United States. So there's no percentile that you could get. The viruses they were interested in, I thought this was interesting. And they were actually looking at the load. Acute bee paralysis virus. Now, if you see these visual signs, health effects that it says, it doesn't mean your bees have that virus. A lot of the viruses, they overlap. If your bees fly out on a day when it's freezing weather but sunny outside, they drop to the ground, trembling and crawling around. It can be they got fooled by the sunshine. I mean, it could be something like that. But because there are so many viruses that are vectored by Varroa, we are looking to see how fast they're spreading. And so this survey, if you go to their website, I bet you could find links to articles that would on why they're looking at this and what these viruses do and how fast they're spreading. So you notice the national prevalence is like 11%. Is it going to let me? Oh, good. On that top one, acute bee paralysis virus, but you don't automatically have that if you see something wrong with your bees. Don't panic, okay? Promise me. And the bees can carry a pretty big viral load, but it's a mix often of these viruses. It's hard to say. And see, Gary is still way low, even though they're talking about millions of viral copies per bee squared. Okay, it sounds like a high figure, but he's still fine. His apiaries, his 8 hives are still fine. And again, on the next one, a newly discovered virus. They're not sure how serious it is. Is it the common cold or is it influenza, latest, most virulent type sort of thing? It's they don't know yet. So they are doing surveillance. So they found if they detected the first two. The third one they didn't detect. And I quite possibly they didn't expect to because they don't have a percentile out here at all. Not NA means oh, they're not they're not looking at it. Okay. But again, a lot of different, and here's that black hairless bees, for example. Deformed wing. There's 2 forms of deformed wing, and they're much more worried about one than the other. And one of them it seems to affect the bees a lot more. Bees can get deformed wing without varroa. They can pass it. And I'm going to draw a blank here on which one. The one that's becoming more common, I think, is the one they're more worried about and they're watching. And see, it's showing up in 95% of the hives. The other one's showing up in 73%. But of hives that show they've got it at all. But still, percentile wise, the one is not serious at all. The other one is interesting. But the hives were clearly healthy. Okay, So I couldn't get all the viruses on one. So here's some more viruses here. IAP, Israeli acute paralysis virus. That one has shown up in the United States in the past somewhat. So that was the national prevalence there, the 22%. So they're still seeing it in 1 out of 4 hives. That although they said they didn't detect it in Gary's at all. So he's one of the 4 out of 5 that doesn't have it. Huh. And then Kashmir bee virus. Not detected again. They're not expecting to find that. 0 nationally, but they're watching for it. I think it has shown up in the past. And then Lake Sinai, okay, 30% of the hives in the United States detected it. Very low load, nothing to worry about. Oh, I'm sorry, I left a block right on top of the next part. Nosema and slow paralysis. I put that in duplicate. So I hope this is making sense. They're looking for stuff. They've seen some of it in the past. They're seeing it in some hives, and they want to know, and this is over the last 5 years too, what the prevalence has been, where it's been appearing. Okay. So it's really important to be reading the notes carefully, because you might be really alarmed when you see millions and billions of copies or something. But no, they're saying that Gary's hives look really good. And it is interesting.
Energy into finding a cure for the varroa mite itself instead of this stuff?
The varroa mite, okay, that's a good discussion topic. This is a good discussion topic, but varroa wasn't a big problem until the viruses showed up, and they think the viruses that they're surveilling for are a large part of why varroa at the beginning people could treat once a year and it wasn't that big. Now the varroa it's growing faster, and the bees are getting sicker from the same amount of varroa that before would not have made them as sick. Does that make sense?
Yes.
It's what they're seeing. I know we wish we could direct getting rid of the varroa. That's a long, involved discussion. And actually, if you use that research database and you put in something like varroa destructor, and then you put in start narrowing it down, you'll get the review articles where they are discussing the problem of how do we get rid of it, and can we ever get rid of it. Apis cerana, the eastern honeybee, never got rid of it. So it learned to live with it. Maybe that's what we want.
Oh.
Maybe you can't get every flea off your cats and dogs if they're going to go outside and play. And with bees, they're going to drift into other hives and such. Maybe you can't, but maybe what you can do is figure out how to live with it. We've never cured the common cold. We learn how to live with it. Take your vitamin C, get your rest, good nutrition, avoid somebody who's actively, and don't actively spread it. If you're coughing and sneezing all over, don't go out. So varroa they're saying we're probably going to have to live with it. Okay, so that's great discussion. I figured this report is going to lead to discussion. I'd be surprised if it didn't. And then I did it larger in case anybody wants to see. This is the only one that looks a little bit concerning, but Gary's hives are fine. And the one virus often won't kill you. I don't know if most people realize that, but I think bees are like us. It takes more than one condition that you get exposed to the common cold or the flu, and darn it, it's when you've just been sick already and you're still recovering and you're still weakened. It's when you're under a lot of stress, when your nutrition is poor, whatever it is, your immune system gets compromised, as we call it. And in bees, we know that it works a lot the same. That's why we study their immune system a lot. If you put in immunity and immune system and such, where I was doing on the PubMed, you get some really interesting articles. That's a fun database to play with, not just to look me up. And you can look anybody up. Okay, I'm sorry, I've got duplicates here. I think I just was trying to make these the right size. Things that they really aren't interested in. I think that's why I made this slide, is not detected, not really high. This one, they're detecting it, but alone by itself, it's not causing problems. So they're surveying for some things, just doing a quick, does it seem to be there? And when they say not detected, they just probably did the quickest. If they did molecular analysis, it was really super quick. Okay, that's really what I had to say. I can show, should I stop the recording at this point, or do people want to ask questions that go on the recording? Anybody want to unmute and put stuff on? Hey, Michael, you want to put some comments on there? Because I didn't show yours, and yours was up on two things, but your hives are okay, right?
Right. Yeah, I was
Oh,
below the national average. I had my mite load was 1.3 mites per 100. The national
mm-hmm.
average is 2.2. And Nosema, I was at zero point 0.2 and the national average is 0.3. So, and then the
But
deformed wing viruses, I was just below national averages on those. Um, but still a little on the high side. The comment I
yeah.
wanted to make on the
Mm-hmm.
pesticides, Bri said they are going to be checking for pesticides. They took wax samples, but
Uh-huh.
it can take up to a year to get the results back. So they were going to send us this sheet first
Mm-hmm.
when they got those results, and then it could be up to a year or so before we actually got a pesticide report.
Mm-hmm.
But they were going to check the wax samples for pesticides.
So yeah, so I really appreciate and you sent yours. I just already had Gary's all entered in and had really been studying it. And I never do as much prep time as I'd like to for some of this stuff, but I thought it was really neat. So if when you get your pesticide report back, and if Gary gets his back, if one of you reminds me, let's do the follow-up to this
Mhm.
and do that. Um, but thanks so much for sharing it because it's the kind of thing you wonder what they did with that citizen science. And I'm sure if you go back, you will get more information and and it wasn't invasive at all. They were really friendly, weren't they? That's what Gary said too. He said he wasn't much of a talker, so they did it pretty fast. But you liked it, didn't you, Michael? I mean, Bree is so nice.
Yeah, Bree is really nice, and she had the gal from Olympia come up with her, the one that is in charge of her funding for this project. So she had her in a bee suit helping out on mine.
Is that
She didn't
Kathy
go.
Bailey or Cap? No.
I can't remember.
Okay. Yeah.
I thought they were they rushed through it. I was have rather they found my queens, set them aside, and then do their sample. But they just find the first brood frame that looks
Uh-huh.
good and they look for the queen and then they start shaking bees. So
Mm-hmm.
I mean, I don't know how many times I've not seen my queen and end up shaking them or something, but so they were a little rough and I was a little cringy for
Did they
not
shake
knowing.
Did they shake into the white Rubbermaid tub? Did they do it the old fashioned?
Yeah,
Okay.
yeah,
Well, at
yeah.
least you would see the queen if you got your
Right,
eyes on them.
right.
Yeah, and
Yeah.
that is the reason for doing the white Rubbermaid tub is if you're not really super sure you saw the queen, you know where she is. Then if anyone looks at scientificbeekeeping.com, Randy Oliver has, I think, the video there. I know it's on YouTube, and he shows how you take the brood frame, shake into the white Rubbermaid tub as Michael's describing. And the good thing about it is the bees that fly out are older. The ones that stay in are the younger ones you're supposed to sample. And against that white, they tend to spread out in a layer of one as they're kind of crawling around. And you would see your queen, we hope.
Yeah.
And that's one of the reasons a lot of beginners say they're more comfortable doing that than the method that I like, where you hold the flat-sided container and you just tickle the backs of the bees, they fall over into the container and you get your half cup that way. Instead of the way Randy Oliver does it and the way Bree and her boss did it is you end up with bees flying all over the place, and that's not really comfortable. And that sets off a lot of alarm pheromone. So if you were going to do that in a whole bunch of hives, you'd be setting it off. So I like the flat-sided, just tickle the backs. You just take this container and go down the sides. Anybody
But it,
else? Yeah, I can turn the recording off if people don't want to ask questions. Okay, maybe I'll do that Cause I'm never sure if people really want to ask questions. And Karen's right. Why is there no money? Well, there is money being spent, but that's a long